Hello everyone, welcome to all in our biology lecture. We are studying chapter number 7, cell division. In this chapter, in our previous lecture, we discussed meiosis. Meiosis includes two phases, meiosis first division or heterotypic division, that is meiosis 1, and second meiotic division, which is called as homotypic division, we called as meiosis second. In that, we study first meiotic division, that is heterotypic division meiosis first. Meiosis first is, in this division, a diploid cell is divided into two haploid cells. This includes following phases, that is Prophase-I, Metaphase-I, Anaphase-I, Telophase-I, and Cytokinesis-I. In that, we study Prophase-I, which again divided into five substages, that is leptotin, zygotin, pachetin, diplotin, and dikinesis. Now today we discuss the remaining phases, that is metaphase-I, anaphase-I, telophase-I, and cytokinesis. So let's begin our lecture. So here the first one, the next phase in meiosis first, that is the metaphase-I. The spindle fibers becomes well developed, the tetrad moves towards the equator, and they orient themselves on the equator in such a way that centromeres of homologous tetrad lies towards the pole and arm towards the equator. Remember, the tetrad lies towards the poles and arms towards the equator. You can see the diagram. In that diagram, you can see the tetrads which having a 4 chromatids. Okay, due to increasing repulsive forces between homologous chromosome, they are ready to separate from each other. The tetrad, due to repulsive forces, they are ready to separate from each other. You can see here recombined chromosomes. There is a two center poles, and in equator region there is a presence of tetrad. That will be seen in metaphase-I. Then anaphase-I. In this phase, homologous chromosomes are pulled away from each other. Homologous chromosomes are pulled away from each other and carried towards opposite pole by spindle apparatus. You can see the spindle apparatus from each pole, which will help to pull away homologous chromosome from each other. This is disjunction. The two sister chromatids of each chromosome do not separate in meiosis first. Remember here, two sister chromatids of each chromosome do not separate in meiosis first. Meiosis first मध्ये ज्या दोन क्रोमॅटिड सिस्टर क्रोमॅटिड्स ज्या असणार आहेत प्रत्येक क्रोमोझोम मधल्या त्या सेपरेट होत नाही. दिस इज रिडक्शनल डिव्हिजन. The sister chromatids of each chromosomes are connected by a common centromer. Both sister chromatids of each chromosomes are now different in terms of genetic content, as one of them has undergone the recombination. Remember here, the next phase is telophase-I. The haploid number of chromosomes after reaching their respective pole become uncoiled and elongated. You can see the diagram. In that particular diagram, you see in telophase-I, the haploid number of chromosome after reaching their respective pole become uncoiled and elongated. The nuclear membrane and the nucleus reappear, and thus two daughter nuclei are formed. In telophase, you can see there is a presence of two daughter nuclei. Okay, then the last phase, that is cytokinesis. We are studying meiosis first. In that, the last phase, that is cytokinesis. After karyokinesis, cytokinesis occur and two haploid cells are formed. After completion of cytokinesis, there is a formation of two haploid cells. In many cases, these daughter cells pass through a short resting phase, which is called as interphase or interkinesis. In some cases, the change of the telophase may not occur. That means the interphase and interkinesis does not occur. The anaphase directly leads to the prophase of meiosis second. So that is about the meiosis first, which includes prophase-I. Again, prophase, they are subdivided into 5 substages: leptotin, zygotin, pachetin, diplotin, and dikinesis. That is the 5 substages of prophase-I. Now in today's lecture, we are knowing about the remaining phases in meiosis first, that is metaphase-I, anaphase-I, then telophase-I, and cytokinesis-I. After completion of cytokinesis, the next stage of meiosis, that is meiosis second, that will be followed. So let's see about the meiosis. Now see second meiotic division, which is called homotypic division. Homo means same, equal, which is called meiosis second. During this division, two haploid cells formed during meiosis first divided further into four haploid cells. Remember here, whatever two haploid cells are formed during meiotic division first, that will be further divided into four haploid cells. This division is similar to mitosis. Most of the time, question asks which meiotic division is similar to mitosis. So, the meiosis second is the division which is similar to mitosis. The daughter cells formed in second meiotic division are similar to their parents. Daughter cells are formed during second meiotic division are similar to their parent cell with respect to the chromosome number found in meiosis 1. That means whatever chromosome number is present in two haploid cells which is formed during meiosis first, that chromosome number is never changed in their daughter cells. That means whatever four cells form in completion of meiosis second. It consists of the following phases: Prophase-II, Metaphase-II, Anaphase-II, Telophase-II, and Cytokinesis-II. So here the first phase in meiosis second, that is Prophase-II. The chromosomes are distinct with two chromatids. Each centrioles divided into two, resulting in this formation of two centrioles which migrate to opposite poles and form asters. Spindle fibers are formed between the centrioles, the nuclear membrane and nucleus disappear. Remember here, the chromosome number are distinct in two chromatids. Each centrioles, again you can see, divides into two, resulting forms asters. You can see the diagrams. After completion of telophase-I, there is a interphase, and after completion of interphase, you can see the prophase-II will be start. In that phase, you can see the nuclear membrane and nucleus disappear. So disappearing nucleus and nuclear membrane, that is the significantly seen in Prophase-II. Now after Prophase-II, you can see the Metaphase-II chromosome gets arranged at the equator. You can see the diagram. The chromosome that will be appear or that will be arranged at the equator, that means the center of the cell. The two chromatids of each chromosomes are separated by the division of centromeres. The two chromosomes or the chromatids of each chromosome, two chromatids of each chromosomes are separated by the division of centromeres. Some spindle fibers are attached to the centromeres. Some spindle fibers are attached to the centromeres and some are arranged end to end between two opposite centrioles. So this is the characteristics of metaphase-II. Metaphase-II. Now see the next, that is Anaphase-II. The separated chromatids of each chromosome becomes daughter chromosome and moves to opposite pole due to the contraction of the spindle fiber attached to the centriole. Whatever spindle fiber attached to the centriole, that will be contract, and due to this the separation of chromatids become happen in this anaphase second. Then telophase-II. During this stage, the daughter chromosomes uncoil. Telophase-II, the daughter chromosomes uncoiled. The nuclear membrane surrounds each group of chromosome. Nuclear membrane surrounds each group of chromosomes. See the diagram. Telophase-II. There is a appearance of nuclear membrane surround each group of chromosome and nucleus reappear. Nucleus reappear in this telophase-II. Hence, this division is called homotypic division. Remember, telophase-II is called homotypic division. Homotypic division. Then see the last, cytokinesis-II. As like cytokinesis-I, you can see in cytokinesis-II, cytokinesis occur after nuclear division. Cytokinesis when happen after completion of nuclear division. Two haploid cells are formed from each haploid cell. Two haploid cells are formed from each haploid cell, and thus in all four haploid daughter cells are formed. Then thus, in all four haploid daughter cells are formed, and these cells undergoes further change to develop into gametes. Further change to develop into gametes. So that is about the meiosis second. You can see whole phases in the diagram. Telophase-I, that is last telophase-I and cytokinesis. This is the last division of meiosis first. After that, you can see the start of meiosis second. We start from prophase-II, then metaphase-II, anaphase, telophase, and lastly the cytokinesis-II. Now, what is the significance of meiosis? The first is meiotic division produce gametes. Remember, meiotic division is happen only in diploid cell at the time of gamete production. So after completion of meiotic division, most of the time gametes are produced. If it is absent, the number of chromosomes would double or quadruple, resulting the formation of monastrocytes, that is abnormal forms. The constant number of chromosomes in a given species occurs generation is maintained by meiosis. Whatever constant number of chromosomes that will be present in species across generation, that will be maintained by meiosis. So that is another important significance of meiosis. Because of crossing over, exchange of genetic material takes place, leading to genetic variation, and which are the raw material for evolution. Remember here, genetic variation is the raw material for evolution, and that is produced due to exchange of genetic material which takes place in crossing over. So that is the significance of meiosis.